Hello learners, welcome once again to our ongoing series on photosynthesis. Having talked about the details of the photoexcitation and the role of light in photosynthesis, let us now go over as to how the two photosystems were discovered in light reaction and what are the different implications of these. Just to recapitulate, there were two stages of photosynthesis that we had established, namely the light and the dark reaction. And of course, the Q10 of both was different, thereby indicating that the light reaction was a pure photochemical change and the dark reaction was a thermochemical process. And as I mentioned earlier, there would be a need to revise this diagram once and over again as we proceed in our discussion. So, that means again we have to first talk about the photoexcitation of chlorophyll and how does it split water and how does it release molecular oxygen as a byproduct and how does it make NADPH and ATP as the ingredients to be given to the dark reaction for the running of Benson-Calvin cycle. So, as I said earlier, the rationale of light reaction was the release or the release of oxygen and manufacture of ATP and NADPH from basic raw materials like photons from sunlight, which we had mentioned in details in the form of an electromagnetic spectrum in our last lecture, and of course, water. So, these three reactions of photoexcitation now need to be looked in details. So, that means there has to be a fallout after photoexcitation when arriving at the high energy levels. The electrons either they are reacting with some ingredient and if not so, they will come back thereby releasing energy in different forms and it would be the photochemical reaction at the metastable state which would be the most useful one. So, that means if we look at a summary diagram of the energy transfer during photosynthesis, one could find that when light falls on different pigment molecules, obviously like you have in a dish antenna at your house for a cable television, you would find that the focal point is in the middle and that you think should be the reaction center and the entire dish would be composed of different types of accessory pigments which will, and of course, the chlorophyll B, which will now absorb the light quanta or the photons of different wavelengths and would transfer the energy of different wavelengths to the reaction center to perform the process of photosynthesis, which you can now easily see in the form of an animation diagram how the chlorophylls are present in the chloroplast and the antenna complex. But then the light when it strikes the antenna complex, it has to be channelized towards the reaction center and you will agree that all the electrons are excited by the light energy of the reaction center. Then we are talking about the photons. Let us now try to quantify this whole statement. We all agree that one photon of light is equal to one electron released. But then how do we answer the question as to how many quanta of light are necessary for evolving one molecule of oxygen? This gives us a thought to learn some equations and terms. The first term is quantum efficiency, which we say is also the kind of quantum requirement and the quantum yield. It can be expressed in both ways. Quantum yield, which is designated by the Greek symbol phi, is measured as the amount of oxygen which is evolved or the amount of carbon dioxide taken up to the number of photons absorbed. I will repeat, the amount of oxygen evolved to the number of photons absorbed. So, that means what would be the number of oxygen molecules which will be released per photon of light. In other words, the efficiency of any light mediated process after the absorption of a photon can be expressed easily. On the other hand, there is another term called as quantum requirement, which is the quantum number and which is 1 by phi. So, now the statement reverses. It refers to the number of photons or the light quanta which are required to release one oxygen molecule or to reduce one CO2 molecule alternatively. All these terms have been designed in accordance with the Einstein's law of photochemical equivalence, which states that in a photoelectric effect, one hit by a photon heads to expulsion of only one electron at a time from the molecule. So, it is actually 1:1 equivalence. So, it has now been found that 4 electrons are needed to be extracted from a water molecule, then only one oxygen molecule would be released. Why is it so? Because actually 4 H atoms are required to reduce one molecule of carbon dioxide by these 2 molecules of water. So, that means when you look at the equation, finally 4 protons plus carbon dioxide would give rise to a molecule of sugar and or at least a component of sugar and one oxygen molecule and one water molecule. That means we can easily then talk about what is the requirement of light quanta. Warburg, another Nobel laureate, performed pioneering work and he initiated these studies and he found that quantum requirement is 4. That means what is the amount of light quanta required to release one molecule of oxygen. Now, this seems to be very extraordinarily efficient process, but then you should realize, learners, biological processes are efficient, but then you cannot achieve 70 to 80% efficiency, because no machine is 100% efficient. There has to be a mechanical advantage and so many other parameters. So, that means how is it that this process is so very efficient to the tune of about 80%? Emerson and Arnold, they then reinvestigated the whole process and came to a conclusion that as I said in the last lecture, that the overall photochemical process is a 2-step reaction and not a single step reaction, because they found that the quantum requirement is 8 to 10 and Warburg was insisting on 4. So, that means this 4 plus 4 is required at 2 different phases. So, that means you drive a single electron by only one photochemical reaction, you require 4 photons. But then if you are having a requirement of 8 to 10, that means you are exciting the chlorophyll molecule twice, which was the simple logic. So, that means maximum photosynthesis, we agree, occurs in red light, because the blue light excitation also now transfers its energy to the other one, and therefore, according to Warburg, the efficiency seems to be 70%, which seems to be very high, as I just mentioned. So, photosynthesis in reality is not so efficient as was shown statistically, and definitely there is a considerable loss of light energy also. So, with the new calculations, what Warburg had mentioned as the efficiency to be 0.25 has now turned out to be 1 by 8, that is 0.125. Now, this seems to be pretty realistic, and we can now account for all the other losses. Now, how did the 2 reactions in photosynthesis came to be discovered? It is a very interesting experiment which was performed by Emerson and his associate Lewis. in the year 1943. They were working on the unicellular green alga chlorella. They exposed chlorella to wavelengths higher than 680 nanometers. That means in the far red region, and they found there was a dramatic drop in the value of phi. That means the quantum yield fell down dramatically. On your screens you can see the graph which shows that the quantum yield drops as the exposure is approaching red and far red. That means the higher ranges of red. Now, this phenomenon was called as red drop. Emerson said that now photosynthesis becomes much less efficient during the red drop. Later, he along with his co-workers also said that if you want, you can remove this red drop, you can abolish it, you can restore higher rate of photosynthesis by giving simultaneously a wavelength which is less than 680 nanometers. That means between 650 and 680. So, that means if these shorter and the higher wavelengths are given simultaneously, there was a sudden increase, a sudden spurt in the rate of photosynthesis, and this enhancement was called in honor of Emerson as Emerson's enhancement effect or simply as Emerson's effect. So, that means surprisingly now Emerson's effect, the quantum yield was 2 to 3 times more than the sum total of rates obtained by the two light beams individually, which means this was also a fundamental experiment which gave an evidence that there are two photosystems which are perhaps working in cooperation in tandem. And obviously, simultaneously, one is working at wavelengths lesser than 680 nanometers and the other one is functioning at greater than 680 nanometers. So, this was a conclusive evidence, but then if the process of light reaction has to continue, it is absolutely imperative that these two systems need to be driven absolutely together, that is simultaneously or at least in very, very rapid succession. So, that means light is absorbed by not one molecule according to Emerson and Arnold, but by a cluster of pigment molecules, and chlorophyll seems to be the center of these molecules. So, the independent pigment molecules, as I said, and I gave you the example of a dish antenna of your house, it is going to collect the wavelengths, different wavelengths, and then is going to concentrate, it is going to funnel these different excited wavelengths to the main reaction center. So, this concept was giving birth now to the modern idea of photosystems. But then came the idea of quantasomes. At least when this functioning is going on, at least there has to be a photosynthetic unit. So, the earlier idea was that 2500 chlorophyll molecules together act as a single unit, and they are responsible for releasing one molecule of oxygen. People like Park and Biggins even saw certain particles in the grana membrane by their freeze fracture studies, and these granules were seen to be composed of about 250 to 300 chlorophyll molecules. But now this concept seems to be pretty unconvincing, because we find that it is not only chlorophyll molecules, or why talk about only chlorophyll molecules, there is a cooperation of so many molecules which are there together. So, it is the quantasome concept is now gone, and instead we are now talking about light harvesting complex. And light harvesting complex would have many antennae, it would have many accessory pigments, and of course, chlorophyll A a certain number of chlorophyll A molecules which are required to function as the reaction center, because the focal point would be the point from where the electrons would be transferred to an electron acceptor. So, looking at the diagram, you would find that or the animation which I showed you earlier, that the different types of antennae are going to be exposed to photons, of course, photons of different radiations, and then from there, since they are so close to each other, this sheer proximity is going to prompt what is called as the radiation-less transfer or resonance transfer. Ultimately, they would reach the reaction center, and then the reaction center would take over from there and perform the other experiments. So, two photosystems are now recognized which have their own reaction centers. As I said earlier, one is less than 680, which is of course now having higher energy, and one is in the higher ranges of red, which is greater than 680 nanometers. Obviously, it has a higher wavelength, so it has low energy. And of course, these are the chlorophyll molecules which are now surrounded by carotenoids and the other accessory pigments. Chlorophyll molecules may have a different absorption maxima. How are these photosystems organized in a membrane? As you can see on your screens, a three-dimensional diagram which talks about the presence of the photosystems and the pair of chlorophyll molecules at the center. They are constituting the reaction center, and the primary molecules which are present in the thylakoid space, and they are collecting the light quanta of different wavelengths. To confirm the whole story, these two photosystems have also been biochemically isolated to find out what are the different ingredients. Photosystem 1 seems to be coming in fractionation in the upper zone. So, naturally it is lighter, and the trap, the final focal point is a chlorophyll A molecule, which is called as P700, because it has an absorption maxima at 700. So, that means this chlorophyll A 700 is also called as the primary trap, and there are 200 light harvesting molecules of chlorophyll A which are of the 683 type. That means this whole system is running at greater than 680 nanometers. And of course, there are a whole lot of carotenoids and chlorophyll B and different types of electron carriers which are present, and various FRS, that is ferredoxin reducing substance. We will find a detailed discussion of this in our subsequent lecture on the Z scheme. And likewise, the photosystem 1, which was the lighter fraction, is important because you should remember it is a strong reductant. That means it reduces NADP to NADPH. On the other hand, it is a weak oxidant and it is rich in iron and copper ions. One very important aspect is, which will help us to understand the Z scheme, is that PS1 can operate independently, and we never mentioned water here. So, that means photosynthesis could also occur in some of the bacteria which are showing just cyclic photophosphorylation, and that means or in a situation where the lower wavelengths are not available. So, that means this can act alone. On the other hand, the photosystem 2 constitutes the lower fraction in fractionation. So, naturally it is heavier, and here the energy trap is made up of chlorophyll A molecule, which is the focal point, which is P680. So, that means nothing can run beyond 680, and then this is surrounded by 200 light harvesting chlorophyll A molecules and carotenoids, but chlorophyll B and carotenoids seem to be in a larger proportion in the photosystem 2. And then it also has an electron donor Z. What is important is, contrary to PS1, PS2 is a very strong oxidant and a weak reductant. So, it is at PS2 that photolysis of water will take place, and naturally learners, wherever the photolysis of water is going to take place, evolution of molecular oxygen would come from there only. So, it is a combination of the strong oxidant nature of PS2 and the strong reduction power of PS1, which together are responsible for the generation of ATP, NADPH, and of course, photolysis of water. So, on your screens you can find a summary of the light harvesting complexes, which we abbreviate as LHCs, and we find that the LHC2 has a major portion of chlorophyll A, and 4 times chlorophyll A is higher in the PS1 or the light harvesting center 1. And it is the sequential arrangement which is now responsible for the formation of NADPH, ATP, and of course, photolysis of water, as I had mentioned earlier. And these antennae, they are packed so closely to each other that they are going to optimize the transfer by inductive resonance. There is hardly any energy loss. Of course, there will be but a minimum energy loss. So, that means there is minimum wastage of energy because of the sheer proximity, and it is the site for the primary photochemical redox reaction or the oxygen reaction. Now, these light harvesting complexes, they have a unique design and they are present on the membrane either in a vectorial fashion or in a fashion which is on both the sides of the membrane. But what is important is, as I gave you the example of a dish antenna, it is they are enjoying a funnel like design, and you can see on your screens that whatever radiation is being now absorbed, which is resulting in the excitation of electrons, it is being channelized, it is being focused in the form of a funnel and finally reaching the energy traps, that is PS1 and PS2, that is the primary reaction centers 1 and primary reaction centers 2 to carry out the processes. So, that means these two systems which we have talked about, as I said, one of the they are all present on the membranes of the grana, and they have a vectorial heterogeneity. What do we mean by this? We mean that one region of the protein is projecting into the stroma and the other into the lumen of the grana. So, that means there are different complexes which are present and they are located at different sites. Just to summarize, if you look at the summary diagram, it very clearly initiates the vectorial arrangement of complexes. The complex 1 and 2, they are towards the lumen, and then the other ones, namely the complex 3 and 4, they are towards the stroma. If you look carefully, the ATPase complex is towards the stroma, which gives us a very clear idea that the proton gradient is going to be formed from the lumen of the grana to the stroma site. And just to summarize the whole story, we have to find a lateral homogeneity also. How are these photosystems located on the grana membrane? It is not random at all. But for understanding this, we have to first talk about the spatial distribution of the grana membranes itself. As you can see on the screens, there are some portions which are naked or exposed, and they are not juxtapositioned to the other membranes. These are called as non-appressed regions, which are shown to you in white. On the other hand, there are certain faces which are facing each other. So, we call it as juxtaposition, and they are facing each other. They are called as the appressed ends of the granum. Now, if you look at the diagram very carefully, you would find that the PS1 complex, you remember learners, PS1 complex is the one where NADPH is going to be produced. They are present on the non-appressed ends mostly, along with the CF not CF1, which is the ATP synthase enzyme. They are present on the non-appressed ends mostly, and also of course on the unstacked membranes. On the other hand, the PS2 complex, that is water breaking or water photolyzing complex reactions or of the PS2, they are present on the appressed ends. So, this is how we also talk about the distribution of these different complexes on the biological membrane. In addition, there are certain mobile elements also which are not fixed, like the plastoquinones, plastocyanins, ferredoxins, which are the iron containing mobile carriers, and they are going to deliver the electrons from the light harvesting center to the light harvesting one, and so on. So, that means their position is certainly not permanent. They are not integral parts of the membrane. They come and go, and they are also responsible for running of the two pigment systems. So, learners, we have now equipped ourselves with all the information to now go on to our next series of lectures, which will be the mechanism of light reaction. How do we have the photophosphorylation, what is reducing power, what is Z scheme, what is cyclic and non-cyclic photophosphorylation, and of course, leading us to the Benson-Calvin cycle, which ultimately produces sugars. So, that would be the focus of attention in our next series of lectures. Thank you.